Hello, everybody. Welcome to my detailed podcast on cell biology and the biology of cancer and cancer treatments. So, if you've decided to open up this podcast, I admire your courage, first of all. It is possible to understand in a very superficial way uh, new cancer treatments and discoveries without going into the details of cell biology. But if you really want a deep and profound understanding of, what's going on in this very active field of biomedical research, you kind of need to understand this stuff, at a deeper level, because cancer is, after all, a cellular disease. It's a disease that occurs when cells and cellular DNA gets messed up. And so to understand, how that happens and what effects it has in the cell and how the treatments target those abnormalities, you really do have to understand cell biology in a fair amount of detail. So I'm going to try to, as quickly as I can, cover enough cell biology here, hopefully in a comprehensible way, to give you really a good basic grasp of what's going on in the field of cancer research and treatment right now. I hope I can be successful at this, but it's difficult. Living things and cells are some of the most complicated things we've ever found in the universe. And in fact, living organisms are the most complicated thing we've ever discovered in the universe. So, understanding how living things work is really an intellectual challenge. And also, to be fair, we don't completely know how living things work. So some of what I talk about, is only partially or incompletely understood as of, this time right now. But I can tell you that we understand a lot more about this now than we did when I was going to college. I took a course in cancer biology, 40 years ago now. And I think I still have the textbook from that college course. And pretty much everything in that textbook is wrong based on our current knowledge. And all of it is also incomplete. It's amazing how many enormous gaps there are because 40 years ago, we just didn't understand cancer very well because we didn't understand cell biology very well. And cancer is a cellular disease. So I'm going to try to get you up to date and up to speed in modern cell biology to the extent that I can, in just a couple of hours of podcasting. I realize it's still going to be incomplete, your knowledge, even if you manage to understand what I'm talking about. But, at least you'll have a fighting chance of, having some grasp of really what's going on now in cancer and cancer treatment research. To start off, we need to talk about cells, what they are, what they're made of, how they're put together, how they work. And cells are just part of the structure of a living thing. So, if we look at, say, a human being as an example living thing, if you dissect a human being and look inside, you can see visible structures that are pretty much the same in most or all human beings. That is, if you cut open 10 human beings, you see 10 structures that look very much the same. And these structures are made of smaller structures, which also look very much the same from person to person. So in a dissected human, the structures you can actually see with your eye, these are called macroscopic structures because you don't need a microscope to see them. You can see them with your eye. The human body is made of organ systems, which are groups of different organs and tissues that work together to perform a critical function for the body. And the organ systems are made of individual organs. Like, for example, this This is the digestive organ system, which has a lot of different organs in it. It has large intestine, it has small intestine, it has stomach, it has the liver, the gallbladder, the pancreas, the esophagus. These are all part of the digestive system. That's the organ system that helps you digest and absorb your food. So you can look at that whole system, but that system is really a group of interacting organs, like the stomach, which interacts with the small intestine and the liver and the gallbladder and the pancreas, etc. So this organ system, the digestive system, is made of these different organs. And the organs are made of microscopic parts, which are parts that we can't see with our eye. We need a microscope to see them, which means we didn't really know that these parts existed until the invention of the microscope, which was in the late 1500s. And it was really in the 1600s and 1700s that we got a good microscopic view of the tissues that make up organs and the cells that make up the tissues and the organelles that make up the cells. So let's talk a little about this microscopic organization here. Your organs, like your stomach, are made of different types of cells. And usually the different types of cells are organized in sheets of cells, layers of cells. And each layer of cells that are of the same type are called a tissue. So, for example, the organ called what we call a stomach is made up of layers of muscle cells, smooth muscle cells, and those are above the layers of epithelial cells inside the organ, which create acid. And then there's also blood vessels and nerves. But the two major tissues of the organ that we call the stomach are the smooth muscle and the epithelial cells. And those are the tissues. Each of those is made of, very similar or identical types of cells arranged together in sheets. The tissues are made up of individual cells. And the cells are made up of what are called organelles. The word organelle actually means little organ, little organs of the cell. So your body has big organs, which are visible to your eye, like the stomach or intestines or the liver. But your organs, your big organs, are made up of cells, which are not visible to the eye. And your cells have inside them what are called organelles, little organs of their own. Now, the little organs of the cell are not the same organs as the big organs of your body. So stomach is made of stomach cells, but stomach cells aren't made of little stomachs. Stomach cells are made of small structures internally, which have different names, like the nucleus, which is where the cell DNA is, and the mitochondria, which creates the energy for the cell, and the lysosomes, which are actually lysosomes are kind of like the stomachs of the cell. They're the digestive organs of the cell. So all of those little structures inside the cell are the little organs of the cell, which we call organelles. And if you put all those cellular organelles together, which includes, by the way, the covering organelle called the membrane, which is like the skin of the cell. Just like skin covers your body, the membrane covers the cell and separates the inside of the cell from the outside. That's also an organelle, the cell membrane. So the cell membrane, the organelles of the cell are made of large molecules that we call macromolecules. Molecule is another word for chemical. These are big chemicals. And chemicals are groups of bonded atoms. So these chemicals are made of groups of bonded atoms. Large chemicals, macromolecules, are made of large groups of bonded atoms, where there might be hundreds of atoms or thousands of atoms in a single chemical, like little cities of atoms. And some of the macromolecules you're probably familiar with by their common names. For example, DNA is a macromolecule. RNA is a macromolecule. Protein is a macromolecule. Lipids, which are biological fats, those are macromolecules. Starches are macromolecules. All of those macromolecules are made of smaller molecules, which are made of tiny little particles called atoms. So these parts here, the molecules and the atoms, are submicroscopic. Normal microscopes will not be able to visualize those. But normal microscopes can visualize the organelles, cells, and tissues. And your eye can visualize your organs, organ systems, and of course your whole body. So we have the macroscopic parts of the body, which are made of microscopic parts of the body, which are made of submicroscopic parts of the body. For simplicity, quote unquote, in our talk here, we're going to stop at the level of the macromolecule. We're not going to really go much below the macromolecule level, so we're going to kind of skip over the atoms and molecules, except to the extent that macromolecules are just large groups of bonded atoms. And there's different kinds that you find in living things like protein and DNA. These macromolecules make up the organelles. For example, the nucleus of a cell is made of DNA, RNA, and lipids, which are big fat molecules, which make up the membrane of the nucleus. And inside that membrane, you have DNA and RNA. Those are the major chemicals of the nucleus. There's also some proteins there. Whereas the membrane of the cell is made mostly of proteins and lipids, large fats. And then other organelles are made of different things. For example, the mitochondria, the energy organelles of the cell, are made of lipids, they're made of proteins, and they also have some of their own DNA and RNA. But it's these big molecules that make up the organelles, which make up the structure of cells. Cells are actually living things. That is, they're the smallest living unit of a body, which means your body, which you probably think of as just this one thing that makes up you, but your body, which is alive, is actually made up of trillions with a T, of these tiny little cells, each of which is alive. So cells are living things the way you are a living thing. In fact, what you are technically is you are a colony of cells. So you are a big living thing that's a colony of smaller living things, which we call cells. And your cells are made up of organelles, which are non-living structures, but each of the organelles does an important function for the cell to help keep the cell alive, just as each of your organs does an important function for you to help keep you alive. And your organelles are all made of these large chemicals like DNA, RNA, and proteins. Okay, so I hope that sort of makes the structure of your body a little clearer. We're going to focus in our talk mostly on cells and organelles and the big molecules. So we're going to, when we talk about cancer and cellular diseases, we're going to focus on the cells, the organelles that the cells are made of, and the large chemicals that make up those organelles. Since in a disease like cancer, which is a cellular disease, where cells start behaving abnormally, the reason that cells are behaving abnormally is because their proteins and organelles are abnormal. And that's due to a defect in the macromolecule called DNA, or defects, I should say, in the DNA macromolecule, which is the instructional chemical of the cell. So the DNA of a cell, which is generally located in the nucleus here, has the instructions for building the proteins and the organelles that make up the structure of the cell. Those are the working parts of the cell, the parts that allow cells to do what they do to keep themselves alive and allow them to reproduce and to grow and to feed and to breathe. And, the reason we say cells are alive is because they do what living things do, which is they consume, they eat, they breathe, they consume gas, oxygen, they consume food, they burn some of that food for energy, they have metabolism, they can reproduce, they can make more cells, they can grow, and a lot of them can move around. So they have some motility. So those are all the things that we associate with life. And because cells can do those things, we say that cells are alive. But cells can only do those things because they have the organelles and chemicals necessary to get them done. Specifically, the proteins and the organelles that are necessary for cells to carry out those functions, like eating and breathing and metabolizing and growing and reproducing. Okay, so when we talk about cancer, we're going to be talking about cells that aren't behaving normally, that aren't functioning right. And the reason they're not functioning right is because their instructions are messed up. That is, the instructions in their DNA. So let's do a little talking about cells and their DNA. And again, this is going to be very important for your understanding of how living things work. So I may have to move this around a bit. But let's talk about the DNA of a cell. DNA is an instruction manual for how to make cells properly so that they work properly. You can think of DNA, one analogy I like to use is that DNA is like a giant recipe book. It literally has tens of thousands of recipes. Biologists sometimes argue about exactly how many recipes are in the DNA, but probably around 50,000 or so. So think of it as a giant recipe book with like 50,000 different recipes. And each recipe in the DNA book is called a gene. So DNA is made up of genes, just like a recipe book is made up of recipes on different pages. And just like different recipes give you instructions for how to make a different meal, different genes contain the instructions for making different vital cell parts. That includes the cell proteins and different parts of cell organelles. And all of these parts are necessary for, proper functioning of one type of cell or another. The cell proteins and organelles are the working parts of the cell, and they're necessary for the cells to carry out their functions. And when cells carry out their functions properly, that means your organs, which are made of cells, are carrying out their functions properly. So for example, if your stomach cells are carrying out their functions properly, then your stomach is doing its function properly. One interesting thing about cells, and we'll circle back to this later, is that every cell in your body, every cell in a single body, has the same DNA instructions, which you might think is weird because, clearly different types of cells in your body look and behave very different. For example, your liver cells have the same DNA instructions as your brain cells do. But liver cells and brain cells don't look alike, and they don't function alike either. They have some functions in common, but a lot of their functions are different. And they certainly, under the microscope, look very different. So how is it possible that different cells in your body have exactly the same instructions for making the same proteins, same organelles? They have the same instructions, but they end up looking and behaving differently. And the answer to that question is actually simpler than you would think. Suppose I give two different chefs the same recipe book. Am I necessarily going to get the same meals from them? And the answer is no. First of all, the chefs aren't going to make all those thousands of different recipes. Each chef is going to pick some recipes that appeal to them. And different chefs will probably pick different recipes out of the book because they find different recipes appealing. So I can give two different chefs the exact same recipe book, but I can get a different set of meals from each one because the different chefs, they choose to use different recipes and not use other recipes. And this is kind of the way genes work in your body. Every cell in your body has the same set of, say, 50,000 genes. But different cells located in different parts of your body use different genes. That is, they activate different genes and they deactivate or silence other genes. So for example, your liver cells may use a few thousand genes, and your brain cells may use a few thousand genes. But of those few thousand genes used in your liver and brain cells, only about 500 of them will be the same genes. And the other couple of thousand genes are going to be different. So of the 50,000 genes you have, one set of 2,000 or 3,000 is used by your liver cells, but a different set of 2,000 or 3,000 is used by your brain cells. And then there's a common set of 500 or 1,000 that's used by both. So the functions are completely different. There are certain things that brain cells can do that liver cells also do. And that's because they do create, the same proteins and organelles from the same genes. At least, 1,000 or so of the same genes. But then there's a couple thousand other genes that are different. That is, that brain cells use that liver cells don't. And then a different set of 2,000 that liver cells use that brain cells don't. So in other words, they're reading different pages in the recipe book. Now, the actual instructions in the genes don't normally change during a person's lifetime. That is, the instructions you inherit from your parents, the ones you're born with, tend to be the instructions you die with. That is, the instructions for making this protein or this organelle or that organelle, they don't tend to change from whatever it was you inherited from your parents. But they can change. It's not common. They don't commonly change, but they can change. They change when there's something causing DNA damage. So remember, these instructions are located on your DNA molecules. DNA molecules are not infinitely strong. They can get damaged. They're just atoms bonded together, and these bonds can be broken by certain things. So if your DNA gets damaged, then that can cause the actual instructions to change. Now, again, that's not a normal thing. It's not something that's supposed to happen, but it can happen, and does with some frequency in some of your cells. And so when there's damaged DNA and that's changed the genetic instructions, that change the instructions in DNA is called mutation. So when our DNA instructions for making a protein or part of an organelle get damaged and messed up, we call that a mutation. Okay, it's a DNA mutation, a change in the DNA instructions. And DNA mutations can lead to certain diseases, which are called mutational diseases. And one of the main mutational diseases is a disease that we call cancer. So cancer is ultimately a mutational disease. It's a cellular disease, but it's caused when certain important genes in the cell are damaged and their instructions are changed or garbled. Now, in order for a cell to turn cancerous, cancer begins with a normal cell that gets some of its critical genes messed up, and then it becomes an abnormal cell. The critical genes that have to be mutated for a normal cell to turn cancerous include genes that control DNA repair. Those are genes that are those are anti-mutation genes, genes that are supposed to fix mutations. When those genes get damaged, when genes that control the cell timing of cell reproduction get damaged, when genes that control cell death get damaged, when genes that control how a cell interacts with the immune system get damaged, when all of those different kinds of genes get damaged, then we can turn a normal cell into a cancerous cell. But you can't turn a normal cell into a cancerous cell just by damaging one gene. You have to damage a bunch of genes. You have to mutate a bunch of genes. And they have to be genes in critical pathways in the cell, like genes that control cell reproduction, genes that control cell death, genes that control the cell's ability to fix its own DNA damage. Those are the genes that have to be mutated to get cancer. You have other genes that control other things. But those genes, if they get mutated, those other genes tend to not cause cancer. So in order to get cancer, there have to be a number of mutations which have to occur in a single cell. And they have to be in these critical genes for these critical cell pathways in order to turn a normal cell cancerous. It is really almost never possible to get all of those mutations accumulating all at once in a cell. So what that means is that cancer develops over time. It doesn't just like happen overnight. It's not like an infection where one day you can be fine, then the next day you're really sick. Cancer is a disease that develops slowly, mutationally, but you have to accumulate a bunch of different mutations before you get cancer. So, typically in a single generation of cells, at most you may accumulate 1 or 2 mutations in some of these critical pathways. And those are usually fixed by the genes that control proteins that can fix DNA damage. So what you'll get is maybe, over many generations of cells, you might get 1 or 2 or 3 of these critical mutations, but it really takes usually 6 or 7 or 8 critical mutations in critical pathways before a cell will actually turn cancerous. So that's going to occur over many, many, many cell generations, which means not generally when you're a young person, usually when you're a much older person. That's why cancer is usually a disease of age. Most people who get cancer don't get it before their 50s or 60s because of all the damage that has to occur in a single cell line before cancer can happen. Now we have some technical words here. For example, the genes that can cause cancer when they're mutated, like genes that code for DNA repair proteins or genes that code for cell reproduction proteins. Those are called oncogenes. An oncogene is a gene that if it is mutated, if it's damaged, it can contribute to the development of cancer. Not every gene is an oncogene. There's a limited number of oncogenes in a single cell. We don't know exactly how many, because within each of these pathways, there's a number of different genes. For example, you've got several different genes that let you repair DNA. And you don't need all of them to be mutated to get cancer. You really just need 1 or 2 of them to be mutated to get cancer. But different cancers can arise from mutations in different DNA repair proteins. So let's say you have 7 DNA repair proteins. If you get mutations in repair protein 1 and 4, that can contribute to cancer, but so can mutations to repair proteins 2 and 3. So there's different ways of getting cancer, different groups of oncogenes that can give you cancer, and they're not all the same. That's why, the cancer in one person can be genetically different than a cancer in a different person, because different mutations in these key pathways, like DNA repair pathways, can lead to the repair pathway failing and can lead to cancer. And if you want an analogy for that, think about like with a car, there's different ways for a car engine to fail. You can have an engine failure in a car, but there isn't just one way an engine fails. There can be a lot of different ways an engine fails. For example, an engine can fail to work right if its fuel intake system is bad, or if its oxygenation system is bad, or if its emission system is bad, or if its if its engine firing system is bad, like the spark plugs, engine ignition system is bad. So, just like there's lots of different ways to get engine failure in a car, there's lots of different ways of getting cancer in a cell, turning a cell cancerous. So, but always it involves mutation in oncogenes. That's something we discovered back in the 1960s and 70s, these oncogenes, and that you had to mutate some oncogenes in order to get a cell to turn cancerous. But since there's a lot of different oncogenes and multiple oncogenes in each pathway, each critical path, there's a lot of different ways of getting cancer, a lot of different combinations of mutations that can give you cancer. So everybody's cancer is genetically unique. That is, even if we get cancer in the same organ, for example, if I have stomach cancer and somebody else has stomach cancer, then my stomach cancer may have been caused by some different mutations than this other person's stomach cancer. Now that's the bad news. So that means, cancer isn't one disease, it's many, many diseases caused by many, many different mutational combinations in oncogenes. But some better news is that there's always going to be mutations in these different families of oncogenes. Like we know that at least one of the mutations is going to be in how the cell interacts with the immune system, and there's going to be another one, at least one or two, that are involved in growth control genes, reproduction control genes, and usually there's at least one that involved in a DNA repair gene. And the other bit of good news is that when two different people's cancer comes from the same primary tissue, like stomach cancer, for example, when it's an epithelial stomach cancer, all epithelial stomach cancers tend to have at least one or two oncogenes that are pretty much the same. So like 80% of people with stomach cancer, epithelial stomach cancer, will have this one particular oncogene mutated. Now they'll also have other oncogenes mutated, but this one that tends to be in common with all stomach cancers, or at least like 80% of them. So what biologists and health biologists and biomedical researchers like to do is they like to try to identify the oncogenes that are common to a particular type of cancer, like stomach cancer, and then develop treatments to target those particular oncogenes. Because the odds are good that if they can get that treatment working in one person with that cancer, it'll work in most of the people with that cancer. Okay, now that brings us to sort of modern cancer therapies, and I'll have to move myself so you can see this last part here. Modern cancer therapies, or what they call targeted therapies or targeted immunotherapies, are treatments that are based on reversing the cellular effects of the oncogenes that are mutated. So first we got to identify oncogenes that are mutated in particular kind of cancer, and then we've got to find ways of reversing the effects of those oncogenes that are activated. So remember, an oncogene is a gene that has an abnormal instruction, so that the protein or the organelle that's being made from that oncogene isn't behaving or functioning normally. It's behaving or functioning abnormally. And that's contributing to the development of the cancer. So part of a cancer treatment is identifying which of these oncogenes is being made in the cancer, and then what abnormality is that creating in the cell function, and then how do we block or reverse the activity of that particular oncogene. Okay, so for example, if we identify a protein that's being made abnormally in a cancer cell, and this protein is an oncogene that controls how quickly a cell reproduces. So the normal version of that protein causes cells to reproduce more slowly, but the abnormal version that's made in the cancer cell causes the cell to reproduce very quickly and form extra cells that we call a tumor. Okay, so how do we treat that? Well, one way to treat it is to identify the protein that's being made abnormally that's causing the cell to reproduce too fast, and then coming up with a chemical that can block that protein, that can shut it off or quiet it down, or even maybe shut off its being made at all. And that will partly reverse the defects that leading that cell to be a cancerous cell. If we can do that, if we can block the activity of that particular protein. Okay, so I know that was a lot. I went through that really fast. A lot to take in. Bottom line, cancer is a cellular disease. Cancer is the result of mutations, that is, defects or abnormalities in the instructions of the DNA of the cell. In specifically DNA instructions for things like cell division and DNA repair and how the cell interacts with the immune system. You have to mess up all the structures of the cell that are involved with those things in order to turn the cell cancerous. There's different kinds of mutations in each of those pathways that can give you cancer, so not all cancers are genetically the same, even if they come from the same tissue in the same person, even. But the good news is that we can identify oncogenes in cells that are cancerous, and then we can design chemicals to target the activity of those oncogenes to stop their abnormal activity, or at least quiet it down some. And that gives us, if we can do that, that gives us what's called a targeted treatment, a treatment that will affect the cancer cell, presumably without affecting normal cells too much, unlike our current chemotherapy treatments, which affect a lot of normal cells as badly as they affect cancer cells. Okay, so this discovery of oncogenes and the activity of oncogenes and ways to reverse the activity of oncogenes or to stop them from working, that's all part of what's called modern targeted cancer therapy. We couldn't do any of that until we knew which genes were messed up in cancer, and we couldn't even know that until we knew that cancer was actually a mutational genetic disease. And we couldn't know that until we knew about DNA. DNA was only discovered in the early 1900s, and genes were only discovered in the mid-1900s. So we couldn't know how to do any of this before, say, 75 years ago, 80 years ago. And we hadn't identified specific oncogenes, cancer-causing genes, until the 1960s and 70s. So now we're talking 50 years ago. So the ability to, treat cancer in a modern way, in a targeted way, has only really been possible in the last 50 years or so. And it's really developed more rapidly in our new age of, oncogenes and our understanding of oncogenes, cancer-causing genes, and in general, how cells use genes to create proteins and cell parts and how they function. So all of that is relatively recent cellular knowledge. And for example, I'm going to give you another another bit of basic biology, and that's DNA here. DNA is actually what's called a double-stranded nucleotide. It's made of two strands of nucleotides that are wrapped around each other. And a single molecule of DNA can be many thousands of nucleotides long, and it can have the instructions for making hundreds of different proteins or cell parts. And each of those proteins or cell parts is called a gene. And in order for the cell to use those instructions to actually build a protein or a cell part, the two DNA strands have to be separated from each other along the length of that gene. So for example, if we want to build a protein from the DNA instructions here on this DNA gene, we have to first separate the two DNA strands, pull them apart along that gene, and then use one of these strands to create a copy of this strand here, which is an RNA copy of the DNA strand. RNA is like a close chemical cousin of DNA. So if we separate these two DNA strands, we can build an RNA strand from one of those DNA strands that's essentially a copy of this other DNA strand here. So we can copy one of these genes onto an RNA molecule. And then that RNA molecule leaves the nucleus, which is where the DNA is, here in the nucleus, and goes to an organelle outside the nucleus called the ribosome. And in the ribosome, that RNA molecule can be used to help the cell put together a chain of amino acids, which is what a protein is. It's a chain of amino acids. So the RNA molecule tells the ribosome how to put together that chain of amino acids with the amino acids in the proper order so that we make a protein that works and does its job. Now, this is where the analogy of DNA as a recipe book works really well. So if we think of DNA as a big recipe book, if I want to make a particular recipe out of that book, we're assuming now an old-fashioned, like literal book you can hold in your hand. Okay? If I want to make a particular recipe from my recipe book, the first thing I have to do is open the book to where that recipe is. Let's say I want to make a chicken meal, which is on page 1001 of my recipe book. So I have to open the book to page 1001. And this separating the two DNA strands here along a gene, that's opening the book. You can think of the separation of the two DNA strands as opening the book of DNA. And where we've opened the book here, that's where we can start copying the recipe. Now, I don't know about you, but my dad used to use recipes a lot. He used to like to make recipes from his Italian recipe book because he was very into Italian food when he was alive. And so he had a big Italian recipe book, and he liked to make recipes out of it. But he would never bring the whole book into the kitchen because the book was enormous. I think I got him that book for one of his birthdays, and it was a huge, large format book. So he never really brought the entire book into the kitchen to make a recipe. What he would do is he would copy the recipe he wanted to make onto like a little index card. And then he would bring the index card into the kitchen, because then he didn't have like a whole huge book taking up a lot of space on the counter. So this is what's actually happening here when a cell wants to make a protein from a particular gene. It first has to separate the two DNA strands along that gene. So it has to open the book of DNA where that gene is located. Different genes are located in different parts of the DNA. And then where the two DNA strands are separated, it has to copy in the form of RNA the instructions for making a protein. And so it builds an RNA molecule which has the instructions for making that protein. And that RNA molecule is like the index card that my dad used to make. That's essentially copying just one recipe out of the recipe book. Because here we're only using mRNA to copy one gene out of the hundreds of genes that are on this DNA molecule. And then we bring that copy of that one gene into the kitchen, which is our ribosome here. And then that gene tells us what ingredients to add and in what order to add them. So in the case of a protein, the ingredients are amino acids, which we need to add in a particular order in order to make a protein that does what we want it to do. So proteins are made of chains of amino acids, which need to be placed in the right order in order to work properly. And the order of those chains of amino acids are specified in the RNA, which has the instructions for building that protein. So when the RNA gets into the ribosome in the cell, that tells the ribosome how to link together amino acids, what order to link them together in to make a protein that works. But keep in mind that the RNA, which had those instructions, came from the DNA. It was built from one of the DNA strands that was separated when we opened up the DNA along a gene. So I know that's complex, and yeah, it's complex. In order to create a protein, one protein from this whole book of proteins, which has recipes for thousands and thousands of proteins, we have to open the book along a particular gene, separate the two strands of DNA along a particular gene, then use one of the separated strands to build an RNA molecule, which is a copy essentially of one of the genes, and then bring that RNA out to the ribosome. That's our kitchen. And then in the ribosome, the RNA is going to specify to the ribosome the order of amino acids that needs to be in that protein in order for the protein to work correctly, to do the job that we need it to do. So the building of the RNA, the copying of DNA onto RNA, that's called transcription. That happens in the nucleus of your cells. And then using the RNA to build a protein in the ribosome is called translation. So there's transcription, creating RNA from DNA, and then there's translation, creating a protein from an RNA instruction. Transcription happens in the nucleus of your cells, translation happens in the ribosomes of your cells, which are outside of the nucleus. When you have mutation in a gene, some of these letters are going to be different. The order of these nucleotides is going to change, and that will change the order of nucleotides in the RNA, which will change the instructions, and that will change over here in the ribosome the order of amino acids in the protein, which will change the function of the protein. Usually for the worse, not for the better. That means it'll make that protein less functional or make it function abnormally in a way that's destructive to the cell. Okay, so those are the basics of the process of using genes in the DNA to create proteins or cell parts in the ribosome. And this is like the most important thing that occurs in the cell, is building its proteins and its cell parts so that the cell can become a functional entity, because it's these proteins and the cell parts that are built here that allow the cell to function, that allow it to feed, to metabolize, to grow, to reproduce, to defend itself, to do all the things that we associate with life. And this process, transcription, translation, is necessary for that process of life. And mutations, alterations in the gene instructions, are usually anti-life, in the sense of they cause the cell to make important proteins abnormally, so that they no longer function normally, which can either make the cell die or unable to perform critical functions, or causes the cell to perform some functions completely abnormally and behave abnormally. And that's usually what occurs when you get cancer, is you have critical proteins and organelles behaving abnormally. But that's due to defects in this process, specifically in the instruction set here on the DNA. Okay, so that's the beginning of our introduction to what's going on with cancer, and cell structure and how cells make their proteins and parts, and what can go wrong in that. So going to get into a little more detail about that in the next podcast, about how mutations can make things go wrong and what can actually cause mutations in DNA. And then about how we can treat proteins and cell parts that are made incorrectly if we know which proteins and cell parts are involved. Okay, so that's what we're going to cover in the second video on the details of cancer biology. Okay, so if you watch these two videos, you'll have a good primer in the details of cancer biology, and then you can watch like the last cancer video, which is modern cancer treatments, and you'll be able to understand it and follow along much, much better, I think. Okay, so I hope you were able to stay with me for at least some of that. I know it's complicated. All right, so see you in part 2 of cell biology.